My name is Chelsey Spriggs, and my lab studies the relationship between viruses and cancer. Viruses are microbes that can cause infection, and there's a debate on whether they're even alive or not because they can't replicate on their own. They have to be inside of a cell to replicate. We're interested in particular in how viruses get to the nucleus, and that's important because that's where virus genome starts to be replicated. And so once that happens, the cell just can go haywire. We study two classes of viruses, oncogenic viruses, which can cause cancer, and oncolytic viruses, which can be used to treat cancer. My lab is studying an oncogenic virus called the Merkel cell polyoma virus. And this virus is fairly common amongst the general population. It causes cancer rarely, but when it causes cancer, it's very aggressive. The name Merkel cell polyoma virus comes from them being able to cause cancer in Merkel cells. But Merkel cell polyomavirus can't actually infect Merkel cells. My lab has already identified some unique features of Merkel cell polyomavirus infection. Unlike most viruses, this virus actually waits for the nuclear envelope to break down during cell division so that it can get into the nucleus. That helps to explain why it can't infect Merkel cells, because Merkel cells don't divide anymore. Knowing that the cell needs to be dividing can help us to figure out what cell the virus is naturally infecting before it causes cancer in the Merkel cells. And maybe we can use that information to design therapeutics against infection in that cell type. The other half of my research program is interested in oncolytic viruses. We're interested in using these oncolytic viruses as alternative therapies for hard-to-treat cancers. There have been hundreds of clinical trials on developing these oncolytic viral therapies for treatment against a variety of different cancers. And unfortunately, we have not been successful thus far in getting many of these through the clinic because we're not really clear on how this infection and the cancer cell death works. My lab is studying the Lu3 parvovirus. So far, we have been able to infect and kill different types of cancer cells with this virus. Right now, my lab is really digging into the different proteins and pathways in our cell that are important for the virus to be able to do this. Being able to understand how these viruses are causing infection in the first place is the best way to be able to prevent them from doing that. And in the other way, if we can understand how these viruses are infecting cancer cells, then we can make that process more efficient so that we can make better therapeutics.